Hi, my name is Anna Daisy Aguirre, and today I will be talking to you about adapting corn to heat stress via carbohydrate metabolism modification. As global population and temperatures rise, there is an increased need for food security. Sweet corn represents a fraction of annually harvested maize and has an estimated production value of over $1 billion. It is considered the fifth most important vegetable crop in the US, and it is estimated that for each degree Celsius that the global mean temperature increases, there is a 7.4% global reduction in maize yield. Climate change is expected to increase the frequency of heat extremes in the summer, which means that we need to produce corn at a level that can meet demand under heat stress conditions. During heat stress, high temperatures speed up the development of corn. This results in a shorter grain fill period, meaning poor and incomplete kernel sets. This leaves growers with unmarketable corn, resulting in a loss of profits. During kernel development, a large fraction of glucose that is incorporated into starch passes through the oxidative pentose phosphate pathway. 6-phosphogluconate dehydrogenase catalyzes the last step of the plastidic oxidative pentose phosphate pathway. This enzyme has 3 isoenzymes. Thermostable PGD1 and PGD2 are in the cytosol, while the labile PGD3 is located to the plastid. It has been shown that the loss of PGD1 and 2 have no effect on seed phenotype. However, the loss of PGD3 results in reduced starch accumulation and disrupts embryo development. This further results in severe defective kernel phenotypes. Thermostable forms of 6-PGDH have been genetically engineered and have been previously shown to provide heat-stable enzyme activity in endosperm ameloplasts. This study aims to measure 6-PGDH activity and WPGD hybrid field corn. By comparing field corn grown under heat stress and ideal temperatures, we can determine if similar activity levels are observed. If heat stability is demonstrated, then growers can be enabled to plant earlier or later in the season and allow corn to be available outside the traditional harvest window, thereby increasing profits and food availability. Currently, I am working on genotyping various samples as transgenic or non-transgenic, using the embryo from the kernel to ensure that we have true positive and negative controls for each event before working on the enzyme activity assays. Transgenic samples showed amplification, while non-transgenic samples did not amplify. Samples must be replaced to serve as transgenic or non-transgenic samples for the enzyme assay. The endosperm is being used to quantify 6-PGDH activity. By first extracting protein from the endosperm, I can quantify the total protein via Bradford assay. Then, I can heat treat samples to remove all endogenous 6-PGDH activity in the plastid. Finally, I can spectrophotometrically measure 6-PGDH activity. Future work will be focused on measuring starch enzymatically to determine how WPGD is affecting the amount of starch available for kernel development. The field trial will be replicated this spring. Thank you for coming to my presentation. I would like to thank the McNair program and the Settles Lab for their mentorship and guidance. I would also like to acknowledge NIFA for funding this work. Thank you very much.